I'm Captain Stephanie Birdie. I am the Pediatric Chief Resident here at the National Capital Consortium Pediatric Residency Program, and I'll be your MC for the rest of the morning. The next speaker I'd like to welcome up to the stage is Dr. Thomas Fleischer. He's the Chief of the Department of Laboratory Medicine and Immunology at the National Institutes of Health.
In a way, we're doing this backwards. My colleague Sergio Rosenzweig is going to talk about genomic applications. And if you are listening to Dan Kastner and his last slide, he pointed out that in FMF, they've been looking at pyrin in terms of function now for 19 years. And the fact of the matter is that the power of unbiased genomic evaluation sequencing is extraordinary, but when one identifies a variation in the gene, the real challenge is translating that variation into a functional abnormality that you are convinced actually underlies a disease. And so I'm going to quickly go through some of the tools available in evaluating the immune system that would be germane in the setting of a defect, an identified defect. Oftentimes, though, these are driven by the clinical circumstances of a patient. And so this is actually a little bit skewed towards infectious presentations associated with immune disorders. But what we've come to recognize is that immune deficiencies are really more than deficiencies, they're dysregulation disorders. And I'm actually been asked to talk at a national meeting on the paradox of autoimmunity and immunodeficiencies. And I actually don't think it's a paradox. It's actually part of the process. If you have immune dysregulation, you may or may not have problems with recurrent infection. It depends on where the abnormality lies. And you may or may not have autoimmunity. And so they are sort of intersecting Venn diagrams, and then the overlap is becoming the rule rather than the exception. So if we talk about evaluating immune function, in vitro immune evaluation, of course, would be the most desirable, being able to look in vitro. And some of what Raffaella talked about may actually be amenable to looking at markers of altered immune responses. So for instance, looking at serum or plasma cytokine levels. Problem is those remain relatively difficult to interpret. So in terms of what has truly been proven, the one area that we can look at from an immunologic perspective is the ability to make antibody responses. And so we simply look at immunoglobulins, we look at specific antibody responses, and of course that's routinely done in the setting of a vaccine. If we move to ex vivo immune function, we can generally focus on cell-specific responses, and it's both quantitative and functional. And so if we talk about quantitative immune evaluation, and you saw a little bit of this in reference to how patients are worked up in Raffaella's talk, we look at lymphocytes, and I should say that we can also look at other cell types. We can look at neutrophils, we can look at monocytes, we can look at eosinophils, we can look at stem cells. It's the hematopoietic stem cell for certain diseases. And the capacity now by flow cytometry to look at multiple different characteristics of a cell at some level allows us to do functional equivalent assessment. So we talk a lot in immunity about naive cells, memory cells, senescent cells. These are markers of cell function. They're not absolute. And what I'm going to touch on, and you actually saw a slide in one of the slides that Raffaella showed, intracellular flow cytometry. This is typically combined with the evaluation of cell surface markers. So we can look at T cells separately from B cells, separately from NK cells. We can look at monocytes, et cetera. We can assess for the presence of specific intracellular proteins. This is most relevant in settings in which there is an absence of protein, that is to say, the underlying genetic abnormality precludes the production of a specific protein as opposed to a dysfunctional protein. And so I've listed a group of proteins that we know cause either a clear immune deficiency, that is, a recurrent history of infections in their clinical phenotype, or in some cases, such as FOXP3, a very significant presence of autoimmunity. We can look at the cell's response ex vivo, response to specific cytokines, and that was actually illustrated in the patient's stat phosphorylation. I'm going to touch on that. We can evaluate cell function focused on the alterations of proteins in response to a specific signal, typically a cytokine. We can look at baseline activation of cells. We now know that in some of the disorders we deal with, they actually have, and that's actually the underpinning of autoinflammatory diseases, their baseline response is markedly elevated. They're responding to something, or they're actually on an autopilot in terms of producing inflammatory products, typically cytokines, and you've heard about IL-1 blockade, TNF blockade, or inhibition with JAK, Janus kinases, that are proteins that actually are involved in the signaling in response to various cytokines. And we can look at different helper cells. So here is a patient that presents with a disease that in many respects sounds like an autoinflammatory disease. They present with a very striking rash. They present with an inflammatory bowel disease that is very much like celiac disease. They have villous atrophy. They may present with diabetes actually at birth, type 1 diabetes, and a host of other disorders. It's called IPEX. And as you can see in this picture, they're missing a cell that has a protein called FOXP3. It's a transcription factor that's absolutely critical to generate regulatory T cells. And in the absence of regulatory T cells, an autoimmune, autoinflammatory disease actually is the outcome. The only effective therapy for this is bone marrow transplant or immune reconstitution with new hematopoietic stem cells. This disease has a very high mortality, and typically it is fatal in infants. So if we move to functional testing, rather than simply describing the presence or absence of cells or the presence or absence of proteins, we can look at proliferation. This is a historical test. It's been around for more than 60 years. And here is simply, you can see in this rather, is there a mouse? Well, it's all right, sorry. Not terribly important, but I think most of you can see that the patient and the control look very different. And this is a defect in terms of T cell responses. Functional testing, intracellular protein phosphorylation. Okay, so it's not critical that you know the specifics, but basically, in order to signal the nucleus to turn on specific genes such as cytokines, there is a process involved within the cytoplasm of the cell that generates a message that goes to the nucleus and turns on genes that are necessary in a normal immune response, but in an immune dysregulation, you actually have excessive turn on. And so this is a simple little picture, and if you look at the difference between the control and the patient, what you can see is that the green tracing is absent. So normally one would expect that stimulation with these two different cytokines would induce a change in this protein STAT3, which is actually linked to a JAK protein. And this particular patient has what we now know is an infantile presentation of inflammatory bowel disease that is severe, unresponsive to normal sorts of immunomodulatory therapy, and this disease also requires bone marrow transplant to control the inflammatory bowel disease. In our lab, we actually do a whole host of different stimuli. Every one of these is a cytokine. You can see IL-2, 4, some of your favorites. And we look for response to these particular stimuli. Now, the fact of the matter is one can actually look at a baseline situation and see whether there's excessive activation of these pathways. Again, the output of these pathways would be additional inflammatory products or cytokines. And what you saw in the picture that Raffaella showed you is utilizing a JAK inhibitor to look to see that you can actually block this pathway. So if there's a particular pathway generating either cytokines that are damaging to the patient, or in the case she was talking about interferonopathies, where interferon signaling is excessive, you can actually look at a particular pharmacologic agent or a biologic to evaluate the potential for that as a mechanism of interfering with this excessive inflammatory response. There's one other area which we focus on a lot in the world of immune deficiencies, and that is these various types of T cells that produce cytokines, and there are now at least 5 or 6 families of helper T cells, Th1, Th2, Th17, Th10. And here is a picture of a difference that's a little hard to appreciate between the control on the left and the patient on the right. We know that this particular pathway is associated with control of Candida on the mucocutaneous surfaces, that is the production of IL-17 and or IL-22. So what can we do beyond what I just showed you? We can look at T cell or NK cell cytotoxicity. You heard about macrophage activation syndrome. There's a group of disorders that are inherited that actually have a familial form of macrophage activation syndrome, familial hemophagocytic lymphohistiocytosis. And these patients have abnormalities in their capacity to generate cytotoxic T cells. So part of the workup for patients that present early in life or present with an EBV virus-induced macrophage activation syndrome is to look at cellular cytotoxicity. We can look at apoptotic processes, either the intrinsic or intracellular induced or extrinsic or extracellular induced pathway. There's a disease called autoimmune lymphoproliferative syndrome in which this is clearly abnormal. There's some other disorders in which the intrinsic pathway is abnormal. You've heard about the inflammasome. There's actually a flow method that can look at, Raffaella, we don't do it, at ask relocalization. One can evaluate, if you're interested in looking at the effect of a gene. Dan talked about transfecting human defect into murine lines. You can actually look at transfection efficiency if you link it to something like green fluorescent protein and understand how many cells you've actually effectively transfected if you're doing ex vivo studies rather than putting them into mice. So there are a number of tests that we have, but the fact of the matter is that what we really, they are really partnered with genomics. And the critical aspect of following through with a newly identified genetic defect, and you heard about trios, you'll hear more about that, is something that is unique to the patient who's affected with a clinical phenotype versus the parents who, in the case of autosomal recessive disorders, are presumably carriers. But the situation gets a little more complicated when you start talking about autosomal dominant disorders. What we've learned is that in many cases these defects have quite variable expression. Presumably the genetic defect has varied expressivity. There are we're all outbred in the sense we're different than mice, and so we all have somewhat different genetic backgrounds, and the impact of these can be quite significant, and we don't fully understand. I think I'll leave it at that, just to say the tools that we have are expanding. I've not really covered some of the much more sophisticated testing that one might pursue to try to nail down that a particular genetic defect. You heard about a whole host of disorders that have been looked at. The ADA2 story really ended up going into a zebrafish and showing that, in fact, there was an impact of that defect on the vasculature, and there actually was the equivalent of a stroke in zebrafish. And so the tools that are at hand now are actually quite remarkable. And really identifying the genetic defect in many respects with next-gen sequencing is the easiest part of the story. And taking that information and translating it into a functional abnormality that will explain the story that's going on in your patient is, I think, the real hard work that follows this genetic story. And I think the Pyron story that you heard from Dan, 19 years after identifying the defect, they're still working on this. It also is the critical aspect in identifying where we should be going therapeutically. And so I think linking genomics, functional testing, clinical phenotype with the objective of generating an appropriate, a logical therapeutic approach is really what this whole story is about. You heard that from Dan, you heard that from Steve Holland. That's where we want to be, to identify the right therapy for the right disease. And the tools for therapeutics are clearly advancing at a remarkable rate right now as well. And I think I'll stop there.
Thank you, Dr. Fleischer. We're going to take a quick departure from our printed schedule for the day and have Dr. Clifton Dalgard, the core director for the American Genomics Center and the director of the core lab for CHIRP, speak next.
All right. So Dr. Fleischer just kind of set the stage for this talk very well, and what I'm going to be talking about, the resources that are now available at USU to NIH and Walter Reed National Naval Medical Center studies and collaborative programs that have already started and well on their way within this collaborative unity of effort. So about 2 years ago, a new vision at USU arose between a collaboration from NHLBI and USU, and really became a program called CHIRP, which has the word collaborative in their name. It's the Collaborative Health Initiative Research Program, but basically it set the stage and foundation to enable large-scale or population-scale biomedical big data studies within USU and Walter Reed, with collaborators from NIH across the street. And the particular challenge at the time is that USU really didn't have a computational biology program. We really didn't have high-performance compute structures or storage area networks that were ready to store large data sets. But over the last 18 months, we've set up the infrastructure to allow for these larger biomedical big data studies. And today we're actually having several collaborations with NIH to perform these population-based precision medicine initiative studies. And so what really entails is the ability to do whole genome sequencing at population scale, store the data, have it readily available to investigators, and share it in a sandbox-mediated method to enable the experience of clinician scientists to interpret the data for their research cohorts. Additionally, it really is meant to have an iterative approach of using this data to go back to patients within DoD MTFs to hopefully improve and tailor therapeutics and decision-making for treatments. And this is kind of the basic model that you see in a lot of precision medicine initiatives, is it really starts at the level of the clinical study and really well deep phenotyping for the patient or the patient cohort, and then perhaps a willingness or a need for the clinician scientist to ask for whole genome sequencing or any type of omics type of platforms, and then to integrate that data, the phenotyping data as well as the omics data, in a big data lake, and use a variety of different methodologies. It's not just one black box leads to an answer. It's a variety of different case-by-case analysis pipelines that may include individual analysis, trios and quads have been mentioned today, as well as population scale analyses that are now enabled because we have hundreds of thousands of exomes at our fingertips, and to look at rare or private variants within trios, generate some sort of functional in silico score to give scientific teams the ability to now functionally work up a variation to see if it's really the molecular etiology for the disease. And so we've been doing this for a little while now. We've set up our infrastructure, and we now have sequenced 2,600 genomes to date. I'm going to let you know that it didn't happen overnight. We really had a great involvement from NIAID to actually allow us to have our first whole genome sequencer at USU through a intra-agency agreement, and that was a Genome Analyzer 2X that came from Ron Germain's lab. From that, we actually really cut our teeth in developing standard operating protocols for genomic and transcriptomic sequencing. And then around October 14th, when we developed the CHIRP initiative, we decided to repurchase infrastructure to allow us to do this at population scale. And what arose from that is a lot of robotics, a lot of computers, and a lot of sequencers. And that's where we're at today, and we're actually operating at a very fast rate. We affectionately term our genomic center the American Genome Center for the 4 bases of DNA, TAGC. What it really encompasses is a laboratory information management system that tracks all samples coming in, essentials them, QCs them, prepares libraries, and then going into a variety of sequencers for transcriptomics, whole exome sequence, and a whole genome sequencing at scale. Coupled with that is the ability to turn that raw sequencing data into annotated data. So we now have a new high-performance compute cluster at USU. It's basically 2,016 computers in a large cluster, and a 3.4 petabyte storage area network that allows us to store tens of thousands of whole genomic sequences and perform analysis on those data types. Really, after we developed this infrastructure, we now have the ability to do about 18,000 whole genomes per year and 20,000 or more transcriptomes per year. We're really not quite at that throughput because not all of our collaborative studies have been set with IRB approval, but we're running at about 270 genomes per week at this time. And really, we're trying to provide state-of-the-art support for DoD investigators, and as well as collaborative efforts through NIH and all the MTFs that want to participate. This is kind of what it looks like. As I said, it's a lot of robots that QC material, prepare libraries, cluster libraries, and eventually ends up into this room, which is the sequencing center. And we, from material receipt, we can library automatically within 1.5 days. We QC that material and we cluster it within a few more hours, and we get them on the sequencer. And once on the sequencer, we can prepare a very deep whole genome sequence in 2.5 days. You often hear that whole genome sequences need a threshold of 30x for coverage. We actually perform on average about 38x at population scale. And we roughly, when we're on full throughput, and we have been on full throughput on some weeks, we do at least 50 genomes per day at least 35x coverage. A very uniform coverage and very excellent for these genomic interrogations. We have, as I mentioned, SOPs, so these are available to all our collaborators, and it really details the minimal input that you need to perform a variety of different modalities between WGS, WES, and transcriptomics. Back in the day, you would need micrograms of material, but now we actually have thresholds as low as 10 nanograms for transcriptomics and about 25 nanograms for whole genome sequencing. So for precious clinical samples or samples that you have sorted or laser capture microdissected, you're still able to get a whole genome sequence from it. What this really enables you to do is look at single nucleotide variation, look at insertions, deletions, and interrogate some other modalities that we find are very, very important in this modern age of genomics, which include copy number variation, structural variation, which are generally more difficult to do with our short read type of sequencing, but it's becoming more and more readily available for interpretation with modern informatics. Complex heterozygosity, allele-specific expression, transversions, insertions are more readily available at whole genome scale. And that's, to give you an example of one of the early success stories we had when we cut our teeth in 2012 with our GA2X that we received from NIAID, there was an interesting study where an investigator at USU, Dr. Andy Snow, with collaboration with Mike Leonardo, Lou Stout, and Helen Su, we're looking at a very specific phenotype called benta, which is B-cell expansion and NF-kappa beta T-cell anergy phenotype. And there's a small number of patients that are seen at NIH. There was one patient that was diagnosed at the age of 13 months, but still did not have a known molecular etiology for the disease at 18 years of age. Now, interestingly, this individual had somewhat a different type of spectrum of benta, presenting with a moderate B-cell lymphocytosis, and the gene associated with benta is known to be CARD11, and they do have molecular diagnostics for CARD11, but they really are focused around a linker domain between the CARD domain and the CC domain, and they were of a PCR, multiplex PCR Sanger sequencing approach, and really didn't cover the full spectrum. Well, Dr. Snow collaborated with us to do combined transcriptomics and whole exome sequencing, and interestingly enough, the RNA-seq data that spanned other domains that are more difficult to access genomically identified a novel mutation, missense mutation, C49Y, that was found in the specific patient. And then that patient's material was sent for traditional Sanger sequencing at a CLIA laboratory and was validated. Since then, 2 other individuals have been identified in France with the same mutation, and functionally, this variation has been confirmed for NF-kappa beta hyperactivation. So it gives you an example of something that's occurred in the immune dysregulation space at USU, and really was a great example how an investigator could hone in to a specific target and find the molecular etiology for this patient with a hypothesis-driven method. Now, we have as you guys know, genomics technology rapidly expands, and there's a variety of different ways now for us to tackle more complex genomics in terms of complex structural variation, compound heterozygosity. And what we now have is the ability to partition solutions and molecules into smaller volumes, and then barcode those molecules, so when we go to sequence them, we can stitch them back, reassemble them, and generate larger phase blocks of genomic data. So now we're able to not just resolve a variant, or because a variant doesn't equal a genotype doesn't really equal a phenotype, but now we're able to build haplotypes by doing these novel methodologies, and we have a state-of-the-art platform which is called the 10x Genomics Chromium Controller, and what this allows us to do is resolve phase blocks of megabases, or 50 megabases depending on the input material, and that really allows us to uncover larger deletions in haplotypes. It allows us to resolve compound heterozygosity, lets us look at phasing, and then with transcriptomics, we could look at allele-specific expression, like Dr. Fleischer was mentioning in the last talk. So these technologies are now accessible, readily available. We have standard SOPs, so if you have an individual that's done WES or WGS before and did not get a definite molecular diagnostic from us, we can now use improved technologies to build better genome assemblies to look to see if there's a genetic determinant for that phenotype. And we have actually used this already. It's very amazing. We think since we've sequenced the genome, we've uncovered all the variation known in the population, but we haven't really looked at it at haplotype-specific levels, and now we're uncovering novel variation, single nucleotide variation, novel structural variation at population scale. And so this gives you an example, and this is a cherry-picked example because it's in oncology, which has a large amount of structural variation. But using these methodologies, we can really take a look at not only the frequency, the type, but identify novel structural variation at population scale by coupling those two methodologies. And so we've covered a new deletion in a gene called LSAM. It's found to be enriched in African American prostate cancer. It's been functionally validated, actually causes dedifferentiation of prostate epithelial tissue. So it's these types of modalities that allow us to uncover new biological significance within our cohorts. I mentioned that we do have SOPs for standard operating material going into our workflows, our robotic workflows, but we also have some non-standard workflows that allow you to use very precious clinical material, even lower than nanogram scale, using microfluidics and low volume input. And so we can do transcriptomics at as under 1 nanogram per microliter. And what this shows here is that despite varying input from 25 nanograms down to less than a nanogram, data for RNA-seq is no different. So what this really allows you to do, if you're doing flow cytometry or flow sorting or any type of immunomagnetic bead enrichment, we can actually library and profile cells as low as 50 cells per sample. And then finally, we know through bulk transcriptomic sequencing that cell types and tissue types are very heterogeneous, and the signal-to-noise ratio when you're performing in a bulk methodology may not lead to enough of a detection capacity for informative capacity in your studies. So what we've really been working on recently is single-cell transcriptomics to uncover the heterogeneity of patient-derived samples, as well as induced pluripotent stem cells derived from patients. And really, it utilizes the same technology where it allows either cells or molecules to be partitioned into very small nanoliter droplets, and then barcoding the material inside the droplet and sequencing. And what you can see here, this is actually a droplet, and there's a single T cell in this droplet. And then we can sequence the transcriptome of that T cell. And so we've been doing this in collaboration with NIH and the Rare Vascular Disease Program with Manfred Boehm. So this is an example of a patient that has Takayasu Arteritis and very extensive lower aortic calcifications. This individual is non-HLA genetically determined, and also interestingly, the initial hypothesis is that this individual was an ACDC phenotype, which is accumulation of calcification due to CD73 deficiency, but it turns out this patient is not filling that genetic diagnosis. And so we're using single-cell transcriptomics, and what's interesting about this individual is they may be mosaic, and additionally, there's only a small amount of subpopulation of their cells that are primed for inflammatory phenotype. And so we can resolve that when we're barcoding tens of thousands of cells. They may have a small phenotype in the order of just 0.2% that are primed, but is sufficient enough to cause a vascular inflammation in this patient. So in summary, we have a variety of state-of-the-art platforms at USU now, ripe for collaboration. Really, I want to thank the leadership from NHLBI as well as the leadership from USU, Dr. Kellerman and Dr. Maddox, for having this vision and supporting our new infrastructure. And I hope to meet you all during the roundtable for any potential collaborations. Thank you.
Thank you, Dr. Dalgard. Next, we'll have Dr. Sergio Rosenwig, the Senior Investigator at the Immunology Service, Department of Laboratory Medicine and Primary Immunodeficiency Clinic at the National Institutes of Health.
So thank you very much. Thank you very much, Dr. Goldachmansky. Thank you very much, Dr. Jones, for the invitation. It's a real honor and privilege to be here. And let me catch up with what Dr. Tom Fleischer was presented before, what we do at the Immunology Service at the NIH. So let me tell you, if you're a hammer, everything looks like a nail. So I was trained as a pediatrician, taking care of patients with primary immunodeficiencies, so everything looks like a primary immunodeficiency to me. So here, this is a definition that we usually use to think about PIDs. PIDs, primary immunodeficiencies, are genetic disorders that produce abnormalities of the immune system, increase susceptibility to infection. But not just that. they increase susceptibility to autoimmune disease, immune dysregulation, allergy, and also cancer. So think about the PAD or the immune system kind of, whenever the immune system doesn't work very well, we cannot fight the aggressions that we get from the outside. But then the immune system gets crazy and starts attacking ourselves. If we find antibodies or T cells, is autoimmunity. If it's the immune system, it's autoinflammatory. Then we get allergies. And also when those T cells and B cells become crazy and they say proliferate like crazy, we have cancer, lymphoma, leukemia. So this is what about PAD is. So it's a dysregulation of the immune system that makes us a lot of different clinical manifestations. So in terms of that, so as everybody has been talking before, most of our disorders are genetic. So even by the definition, and actually PIDs were kind of 150 disease until 2007. The first one was described as Walter Reed. So the first PID was described by the kennel here, and not this building, the other building at Georgia Avenue. So it was Bruton disease. And now, so we used to have 150 of those diseases. Now we have kind of 300, and this is because of the new technologies that we are using in order to diagnose. And as Dr. Fleischer said and Dr. Dulger said, so we are using a lot of genetics and genomics in order to put a name or a gene to correlate with a clinical syndrome. So whenever we made a clinical diagnosis or whenever we think about primary immunodeficiencies, we use different kind of approaches. So one that we doctors used to use is a kind of a clinical definition. So we said that patients have severe combined immune deficiency. We used to say that they have velocardiofacial syndrome. So we use a clinical approach to diagnose them. But then we can also use immunological approaches. So they can have A-gamma globulinemia. That means that they have no IgG, IgA, IgM, IgE, and IgD. Or they can have hyper, as we heard before, hyper-IgD, hyper-IgM. So we have disorders of low and high aspects of the immune system. And then we can also define them, as Dr. Holland said before, because of their susceptibility. People, they are susceptible to mycobacterial diseases, or they are susceptible to herpes infections, or they are susceptible to candida infections. So not all of us are susceptible to the same kind of microbes. So we can all be exposed to them, but just a certain group of us are going to get infected and sick because of certain type of microorganisms. So what that means is there is some kind of genetic heterogeneity. So it could be, or we can have multiple gene defects that could be associated with each of those phenotypes that we have just described. So as an example of that, so we have patients, I told you, I am a hammer, so everything looks like a PID to me. So everything is primary immunodeficiency. Sorry, Dan, sorry, Rafaela, including the autoinflammatory syndromes, you're under our umbrella. And because I am at the podium, I can say whatever I want, and then you can come here and say whatever you want. So here, severe combined immune deficiency, you can have 13 different gene defects that they're going to present very similarly. Same thing happened with people that they have no B cells and they have no immunoglobulins. You have different defects that they're going to all present with a rest in the maturation of B cells, and they will have no immunoglobulins. And very similar things happen, as Steve Holland said before, multiple gene defects can give you susceptibility to mycobacterial diseases. So that means that we have one clinical phenotype, multiple gene defects that they can give you or associate with that clinical manifestation. And that's why we were moved into a kind of a broader kind of approach in order to make the diagnosis. So you already heard about whole genome sequence, WGS, whole exome sequence, or targeted NGS approach. So all of them, they are talking about the umbrella of massively parallel sequencing or next-generation sequence. So there are different techniques, different approaches, just to make us done very simpler. So we don't know what the patient has. We have a clinical phenotype, but let's look at all the genes at the same time, because there are multiple genes that they can provide a single clinical phenotype. So here's what we are talking about in terms of numbers. So when we are talking about the genome, we are talking about 10 to 10 to the 9 coding and non-coding base pairs. We are talking about the exome. We are just talking about 1% of that genetic information. So here we have 3 to 10 to the 7 coding base pairs divided in 180,000 exons, 20,000 proteins, 3.5 million single nucleotide variants, 10,000 non-synonymous variants, 50 to 100 heterozygous mutations. So my mom and my grandma, they thought I was perfect. I'm sorry to disappoint you, mom and grandma. I do have mutations, as all of you have. So it doesn't mean that we are sick because of that, but we are all full of mutations in our genome. But some of us are more lucky than some others of the patients that we have seen here. So our mutations perhaps didn't manifest yet, or they will never manifest. But our patients, they have the bad luck to have the mutations and have clinical manifestations associated to it. So here's how it works. So there are multiple platforms. You heard Dr. Dulger talking about very complex and sophisticated. Let me try to make it in a way that I can understand it. So here you have DNA, very small amount of DNA. Dr. Dulger was talking about nanograms. That's tiny, tiny, tiny amounts of DNA. And then you chop it mechanically or using restriction enzymes. Then you hybridize. You make it match with another pieces of DNA. Then you get rid of whatever was not hybridized. You capture the DNA using different methods. Then you have a magic machine here. And then you have here the ta-da effect or the hocus pocus, and then the magic, and then you have the results. So this is how it works, at least in my mind. So there are multiple platforms, there are different instruments, but all of them work in a similar way. So then we'll do this kind of magic, so the ta-da, the hocus pocus effect. So here you have, we're going to run the metrics. Then we're going to do the alignment, variant calling, and annotation, and everything going to be kind of fun and dandy. And this is where the money is, because running those tests now is not that expensive. Doing the analysis is where the money is, because this is what really need a mind, an intellect, somebody with a brain in order to make the interpretation or whatever the machine gives us. And then we have the IGV, Integrative Genomic Viewer. That's a way of looking at the mutations. At the end, you have some kind of result. But then everything going to come back to your patient, and you have to put together. You have a clinical phenotype, you have a mutation. As Dr. Fleischer said, that's the easy part. Now you have to connect them. So Dan Kaslan said that he saw the patient in '85, he found the mutation a couple of years later, and it took him years in order to make the connection between the two things. Because I told you, we think that we are perfect. We are not. We are full of mutations. Making that connection is what it can take you years of your life in order to connect one thing to the other one. So here, molecular approach at the NIH, at the Immunology Service, we do whole exome sequence with targeted analysis. We analyze 350 prime immunodeficiency and autoinflammatory genes known to produce diseases, and then we do also the Sankar sequencing validation and family segregation of the mutations. As Dr. Fleischer already displayed and already explained to you, we do also the flow approach. So these include basic panels that to discriminate between T cells, B cells, and NK cells. We do complex panels. They're all upon approval of myself, so you have to be nice to me if you want them to be approved. So, and then we do multiple different things. One thing that you have to keep in mind, so we are a research institution, so every patient that we're going to test, it has to be enrolled in one of our, enrolled and consented in one of our research protocols, and that's very easy to be done. It can be done over the phone, but we cannot process samples just right away before just consenting the patients. So you're in the military, you understand we have our own SOPs, so we have to follow the rules. Patients cannot be tested just right away. They have to be consented in order to be tested. So here, the senior staff, so we are the ones that we take the credit. Here's the people that really does the job. So Jennifer Storter and Julie Nimela. So all our genome and genomics approach is 1.5 FTEs. So all what we do that they're between 500 and 600. I know that sounds nothing compared to that you heard before, but for us, with a very small operation, we're pretty happy with what we do. So Jen and Julie do the molecular approach, the flow cytometry by Kim, Jude, and Shakuntala, and as I told you, we're gonna take all the credit for what they do. Thank you very much.
Thank you, Dr. Rosenwig. Next, we'll have Colonel Peter Weina, the Chief of the Department of Research Programs here at Walter Reed National Medical Military Center, to speak about our research labs and our graduate medical education research.
Well, thanks for the invitation to come and speak to you about what we do here in research programs. And for those that don't recognize this is Building 17B, or 17A and 17B. Our program is actually, nobody knows where it's at, so I'll tell you, it's where the gym is. It's where the pool is, okay? So if you walk in there and you find the gym and you find the pool, instead of going on the first floor, just go to the third floor and you've got us. So we're there. This is actually one of the best kept secrets around here. We actually provide, rather than just, a lot of people think about us as just the regulatory police. And one of our jobs is to protect human subjects that are involved in research programs. But really, the way we look at our mission is that our job is actually to support the research mission here. And while we may not always be able to say yes, I've kind of trained my people over the last couple of years to not say no, but to find a way to say yes. They may not be able to say exactly yes to your exact question, but they'll find a way of helping do that. And they do that by providing technical guidance and support. They actually help people design the protocols. So instead of trying to find 75 different parameters for every single project that you have, so you go from basic research answer to a Nobel Prize and never finish the protocol, to actually doing protocols that are actually executable and can be done in the period of time that you have planned. We also have on the fourth floor of this building, above the gym and above the pool, we have advanced instrumentation in a 12,000 square foot laboratory. And if you've never been up there, we have a lot of great gene whacking and cell smashing toys, and we have support staff that can help you with that. And we also train individuals and get people, kind of whet their appetite as to what actually is available in there. And we have short courses in immunology. We have basic laboratory courses that we offer. And my staff have always been wonderful as far as jumping in. And if somebody is just interested in seeing what's up there, they're all very happy to take you through and show you what is capable, what we're capable of doing and what we're not capable of doing. And if we're not, quite a few of our people actually have great collaborations throughout NIH, rare uses, a variety of different places. In just the last fiscal year, we actually supported 7 externally funded grants and 5 GME projects, and I'll come back to those GME projects in a moment. As I said, we offer these laboratories, training. We train physicians, clinical staff, the nurses, and this one was just a recent example of that. The laboratory itself is actually fairly well staffed. We have military as well as GS and some contractors, and we have chemists, we have physiologists, we have immunologists, and we have a variety of different individuals that can help you get things done. Our capabilities are kind of listed here. The main thing I think that we provide is assistance in generating, not just generating the data, but helping you get the ideas, helping you fit what you want to do into the capabilities that we have. As a matter of fact, when I came here, I've always had this kind of dream. It actually goes back to 2002 to set up a new type of diagnostic. Rather than knowing a disease and then testing specifically for that disease, a way of approaching it so that we can just say, well, this person's sick, what's wrong with them? Kind of the Star Trek tricorder type idea. And we actually are working toward that end. But you can see we've got a lot of things in molecular biology, immunology, analytic chemistry. Our chief right now, unlike a lot of our previous chiefs, our chief right now is actually an inorganic chemist, and he's doing some very interesting things with diagnostics with inorganic chemistry. We also, other resources that we have within the department that people don't know about is, one of them is funding. When I first came here, the idea is that somebody told us that we had $250,000 somewhere for graduate medical education funding. Turned out that was totally fictitious. Nobody had any idea where that money was, and when I went to the comptroller's office, they said, yeah, that's a nice idea, but it just doesn't exist. So 2 years ago, we actually had $0 for GME research. Since that time, we've actually managed to generate over $1 million in this last fiscal year for graduate medical education, and we've given out a tremendous number of grants. The old value was about $7.5K for per year, but now I think we can start increasing that, and we're starting to look at anywhere from 10 to 20K per projects for graduate medical education research. So we actually have grant managers that help people actually find outside money as well. And on a bet, we actually went ahead and got $300,000 to start another project just very recently. So, and just in the last fiscal year, our scientists have generated several $1 million of research money coming into our laboratory. We also have statisticians, and these are not the theoretical statisticians that sit and bore you to death. They actually help design projects. They actually help analyze the projects, and they help with writing the papers so that it makes sense. And we have 4 of them, and they're all exceedingly good at it. We have protocol analysts that we used to call them protocol navigators to help, and I think that's the NIH term, but help people get their protocols into shape, help them get into our new EIRB system, and those that know the problems associated with that recently. Our teams are standing by to help so that you don't feel like shoving a sharp stick through the center of your forehead, and you actually can get a protocol through. And then we have a business office, and for those of you that have been in research long enough, you know that research is a contact sport, and it does take money. We now have a business office that actually knows how to get money and how to set up collaborations and how to make sure that we have the documents in place to bring the money in, and to make sure that the work is adequately supported so that you don't get partway through the project and go, oh, geez, I don't have any money. So it's a good group, and it's a resource that's available for graduate medical education as well as for other researchers within the institution. So, thanks.
Thank you, sir. Our next speaker will be Colonel Thomas Baker, Senior Pathologist at the Joint Pathology Center of the Defense Health Agency.
So it's retired, by the way, so. Okay, anyways, thank you for the opportunity to come and talk today. What I wanted to talk about, I wanted to talk a little bit about the Joint Pathology Center. How many people here know what the Joint Pathology Center is? I know Dr. Horkane does. So I want to talk a little bit about who we are and what are some of the opportunities. For those of you who don't know, the Joint Pathology Center was established in the National Defense Authorization Act of 2008 to serve as a pathology reference center for the federal government. And the intent was to replace several critical services provided by the AFIP, which were identified for closure under BRAC, Base Realignment and Closure Act of 2005. If you look at NDA 2008, you'll see that there are 4 missions for the Joint Pathology Center. One is pathology consultation, second is research, third is education, and then the fourth, and this is the one I really kind of want to focus on today, is management and utilization of the former AFIP tissue repository. And we'll talk a little bit about what that is. And really to kind of understand, how the Joint Pathology Center came about, you need to have an understanding of its background. The Joint Pathology Center opened its doors in April of 2011, And as I said, we were formed as a result of the National Defense Authorization Act of 2008. And once again, that was in response to the AFIP, the Armed Forces Institute of Pathology, closing in 2000, or identified for closure in 2005. And so if you understand what the AFIP was, you'll understand what the JPC is. The AFIP was actually formed initially during the Civil War as the Army Medical Museum, and with time, the Army Medical Museum took missions that were needed by the Department of Defense, in the area of the evolving area of pathology. Pathology was still in its infancy back then, and so there were not a lot of resources out there, and the Army Medical Museum basically provided those efforts. And so the Army Medical Museum over time transformed into the Armed Forces Institute of Pathology. And if you look at what the Armed Forces Institute of Pathology did, they had a pretty significant and tremendous clinical mission for pathology consultation that extended actually across the world. They served not only Department of Defense, VA, they also served civilian facilities, and they served underserved communities overseas, so they had a global outreach piece. And then the research piece was also very extensive as well, and we'll talk a little bit about that. So in its heyday, the AFIP was known for not only its clinical, its global outreach, but also for its research. And so historically, you go back and you see names in pathology, and a lot of the big names were from the AFIP. And if you look at historically at articles, you'll see the AFIP as being a key factor in a lot of the research that occurred back through the 40s, 50s, 60s, and 70s. As I said, the AFIP was slated for closure in 2005, and then ultimately closed their doors in 2011. The Joint Pathology Center, as a result of the NDA 2008, ended up taking a handful of its missions. So the AFIP was pretty expansive. We were small and lean, which was expected at the time. So when we opened our doors in April of 2011, our initial focus was on the clinical mission, and we'll talk a little bit about that clinical mission. And unlike the AFIP, which had a lot of external funding, we were funded solely by the Department of Defense. And even though our mission is to support serve as the Pathology Reference Center for the federal government, the funding comes from DoD. And so what that means is that services that we provide to other federal agencies are through formal support and are reimbursable, and we do that with the VA. Once we've matured our clinical mission, we started focusing on our other missions, research, education, and management of the tissue repository, and we'll talk a little bit about those. And our priorities are largely aligned with the military healthcare system strategic priorities or research priorities, and so you'll see that as you get a better understanding of the Joint Pathology Center. And then, of course, and this has been discussed a little bit today, we're aligning with and integrating into the developing academic health system concept with Walter Reed and USIS, and then across the street with NIH. So this, I think, is actually a perfect time to talk about the JPC as a part of this effort moving forward. So let's touch a little bit on the clinical service. Our clinical service really is focused on secondary subspecialty pathology consultation. And so you think about what secondary subspecialty pathology consultation means. These are services that are provided to pathology departments in hospitals. We serve all of the DoD medical treatment facilities, and through agreement, we also provide services to VA hospitals and clinics, and that amounts to about 235 facilities that we serve. We also do some other work as well, but the majority of it is to the DoD and the VA. We provide subspecialty consultation in all areas. We have 30 pathologists. Many of them are well recognized in their fields, but they're all subspecialists, and their full-time job is to provide subspecialty consultation. We also have supporting histology laboratories as well as a molecular laboratory. We have the Department of Defense's only neuromuscular laboratory. And then we have transmission and scanning electron microscopy. And for those of you who aren't familiar with scanning electron microscopy, we have one of the few clinical labs in the United States that uses SEM or scanning electron microscopy as a clinical tool. And then we also have a clinical laboratory for heavy metals detection and quantification. This is largely to support a VA program for depleted uranium follow-up. So, you know, as with any clinical service, our subspecialty consultative service really requires, you know, for us to grow and to mature as a consultative service, we need to develop relationships outside of the Joint Pathology Center, and that's what we've been focused on for the last 5 years, including relationships with Walter Reed, with USIS. We have some relationships with Department of Pathology at the NIH, as well as outside of the federal government. Our molecular capabilities, having state-of-the-art molecular capabilities is key for us to be able to do our business, and so we're working on bringing that to where it needs to be. It's not exactly where it needs to be right now, but bring it to where it needs to be, and then making sure that it supports the mission. And so research, education, and the tissue repository, and we're going to really focus on the tissue repository as a resource for research. The tissue repository is a centerpiece of our research and education mission. And so our tissue repository consists of material, and so we actually have material embedded in paraffin collected through the consultative service from 1917 to now, and so it's almost 100 years worth of tissue, and it's really the largest collection of its kind. It represents 7.2 million patients, 32 million tissue specimens, 55 million glass slides, and so when you look at it, it really represents the largest collection of disease cohorts in the world, and I think beneficial, and one of the opportunities here is it's a fairly large collection of rare and unique specimens. So, you know, when you think outside of the common diseases, there is opportunity here. Our tissue repository is, we talk about it as being a biobank, but it's very, very different from the biobanks that you think of in your institutions. Biobanks tend to be consented biorepositories with clinical information. They have tissue specimens that are collected under very precise circumstances, very controlled circumstances. They may or may not have serum specimens and other bodily fluids. Our collection contains only the material that was collected during clinical care, for example, a biopsy or colon resection or things like that. And so they're embedded in paraffin, and so they were not collected under controlled circumstances. They do not have serum specimens that go with it. The patients were not consented for use of the material for clinical research. And then lastly, you look at the data that goes with it. Not a lot of clinical data oftentimes. So it's very different from what we think of as a biorepository. You know, that creates challenges, but also creates opportunities that perhaps you don't see with other biorepositories. And the Joint Pathology Center, our goal with our tissue repository is really to broadly open it up for use in research and education in order to advance medical knowledge. Once again, that's our goal. We have a lot of challenges to really do that on a wide or broad basis, but it's where we want to move to. We did contract with the Institute of Medicine to review and make recommendations regarding our tissue repository and really how to open it up. And they released a report in October of 2012, and we've kind of used that as a basis for starting to develop our tissue repository. And the Institute of Medicine looked at not only operational issues, how could we do this operationally, but also looked at the ethical issues regarding uses of the specimens in the repository, given that, you know, our repository is nobody was consented for use of the material in research. So they provided us a way forward on this. And that's the basis for what we're doing with our tissue repository these days. As I said, our tissue repository, very different from a biorepository. We have significant challenges that we're working to address, and a lot of this, of course, requires resourcing. And so we've been pursuing resourcing to be able to address these challenges. And so a couple of significant challenges. Once again, the material that we have in our tissue repository on 7.2 million patients. These patients were not consented for use in research. Our clinical information is oftentimes very limited, and follow-up is either non-existent or not limited, or not available. We have, since our material goes back to 1917, it occupies 42,000 square feet of space up on the Forest Glen campus, and if you walk in there, you'll see why it's, why it can be a challenge to actually find the material. We have challenges with querying repository materials for the, or the repository for cases. We have multiple legacy databases that are sometimes very challenging to query. And then, of course, specimen integrity issue. And once again, you compare us to a true biobank, and they have very formal way of obtaining tissue to minimize all the pre-analytic variables and things like that. We don't. We get these things in the mail. We have no idea how they were collected. We have no idea how they were stored till we get them, and we have no idea how stuff back in the 70s was stored at the old AFIP. So a lot of specimen integrity issues that, as we use these for research, we address these at that time. And then, of course, as an organization, we had a lot of requests for specimens, but we have to ensure sustainability of the holdings and make sure that it's a valuable asset for years to come. And so that's one of the things that we're addressing as well. So I think that's basically it. Just to give you an idea, and once again, keep this in mind, when we talk about future opportunities with this program and similar programs. What are the opportunities for the Joint Pathology Center to be a partner in that? Thank you.
Thank you, sir. Our next speaker is Colonel Thomas Newton, the Chief of the Department of Pediatrics and the Children's Center at Walter Reed National Military Medical Center.
So hopefully after our talk on the Children's Center, you guys will get a new appreciation for what pediatrics is over here at Walter Reed. Prior to taking my current position, I was the Chief of Children's Oncology here for 4 years, and as part of that position, we are part of the Murtha Cancer Center, and we get a lot of DVs that come through and visit, congressmen, other important people, and the first question we always got was, wow, we didn't realize that there was pediatrics here, and more importantly, that they were very surprised at the capabilities that we had here at Walter Reed. So this slide is to show you that Walter Reed is under a very special category just in terms of how we fall into the Department of Defense. We do not fall under any of the services. Most military treatment facilities either fall under the Air Force, the Army, or the Navy, and we fall under none of those. Walter Reed, as well as Fort Belvoir Community Hospital down at Fort Belvoir, Virginia, fall directly under the National Capital Region Medical Directorate, and we report directly to the DHA. So we're a little bit unique in terms of our medical reporting chain. We work together with all the other military treatment facilities here in the National Capital Region to provide global health care to the children in the National Capital Region, as well as we're a major referral source for children primarily in Europe and other places along the East Coast that don't have close access to specialty care. There are about 200,000 local children that we care for in both our primary care and our subspecialty services. We partner a lot with Fort Belvoir Community Hospital. As most people here are from the National Capital Region, you realize that traffic is a small problem we encounter here in the National Capital Region. We recognize that that is very frustrating to our patients, and most of our subspecialists have pretty significant footprints in terms of clinical practices down at Fort Belvoir, so that our beneficiaries that live down in Virginia don't have to make the trek all the way around the Beltway to come and see us. We fall under the Directorate for Medical Services along with the Emergency Department, Neurology, and Internal Medicine here at Walter Reed. Our history. The Department of Pediatrics has been around for a long time. We were founded in 1949 as a pediatric section under Colonel Ogden Bruton. He had been mentioned earlier as one of the pediatrician that identified Group Bruton's A-gamma globulinemia, and our inpatient unit still carries his name. We're a fairly large department, and as I hope you'll get to see, we really have the capabilities of a small to medium-sized children's hospital here at Walter Reed. So we're a hospital within a hospital. We have board-certified specialists and subspecialists in many different fields. We have about 235 personnel, physicians, nursing staff, support staff assigned here directly at Walter Reed. And in addition, the Children's Center is made up of another 36 faculty, researchers, and staff at the Uniformed Services University that all come together to provide the care that we do here at Walter Reed. We see over 30,000 primary care visits and over 25,000 specialty visits here, and another 5,000 to 10,000 at the military treatment facilities in the National Capital Region. This is a little busy, but as most slides show, our department is fairly complicated. In addition to the administrative support that falls directly under my executive office, which includes our quality and process improvement, we have broken up our department into 6 major services, and that's where all of our specialties fall under. We have a primary care service, an adolescent service, a hospital-based service for our inpatient services, a hematology-oncology service, since that's kind of a unique subspecialty, and then our other subspecialty services fall under the generic subspecialty service. Our primary care medical services here, we have 4 primary care medical homes, just like most military treatment facilities in much of the nation has gone to adopt the primary care medical home as the patient-friendly way and the right way to provide primary care to our beneficiaries. We have done the same. Just this past year, we got NCQA Level 2 certification as primary medical homes for our 3 pediatric and our adolescent medical homes. Within our medical homes, we have pediatric nutritional services, integrated pediatric behavioral health, lactation support, and social work support. We have small inpatient pediatric services here as well. We have got a 14-bed inpatient multi-service unit. That unit is both medical and surgical. It houses anything from our general pediatric patients to our oncology patients to our post-op neurosurgical patients. And it is staffed with a fully credentialed pediatric nursing support. We have a small pediatric intensive care unit, as well as a good-sized neonatal intensive care unit, and then our mother-baby unit. One of the special parts of our inpatient services is our pediatric sedation unit, where we're able to provide outpatient sedation support for procedures, such as perhaps bone marrows for oncology, as well as for radiology support, so that we can do MRIs and long radiology studies on even very young infants here. We have a large number of pediatric medical specialties, and most of our specialties are 2 to 3 deep in terms of our number of subspecialists, so we're really able to provide a large breadth of services among them. Some of our special areas are child abuse. We house the Armed Forces Center for Children Protection here. So in addition to providing specialty consultation for the National Capital Region for cases of concern for abuse or neglect, our specialists go all over the Department of Defense to provide consultation. They act as expert witnesses and to provide training at other bases across the United States. But as you can see, we have a large number and pretty much all of the subspecialties medically that you'd find at a children's hospital. In addition to providing clinical care, we have a large amount of research that goes on in our medical subspecialties. In addition to the 30 to 40 children's oncology group open protocols that we have here at Walter Reed, there are another 2 to 3 dozen additional clinical protocols spread throughout the different medical specialties that are going on here at Walter Reed. We also have a large variety of pediatric surgical specialties, everything from orthopedic oncology to pediatric neurosurgery to pediatric plastic surgery and craniofacial reconstruction. We're able to provide virtually all of the surgical needs for children here at the Children's Center at Walter Reed. We also do a large amount of education here. We have the Department of Defense's largest pediatric residency and the only tri-service residency with Air Force, Navy, and Army residents. And we have 6 fellowship training platforms here: neonatology, endocrinology, gastroenterology, hemonc, infectious disease, and neurology. In addition to that, we're one of the primary teaching sites for the clerkship or the 3rd-year rotations at the Uniformed Services University, and have a large number of 4th-year and sub-I students that come through, primarily through the summer and fall, who are coming to do their rotations here in hopes that they'll join our residency program the following year. So I hope you can see we have a good footprint here. We are really working to change our branding, as we'll say, from the Department of Pediatrics to really let people know that we are the Children's Center here at Walter Reed, and we really offer the full breadth of medical and surgical specialties that one would expect to find in a small children's hospital. Thank you.